Okay. All right, good. Yeah, so good afternoon, everyone. So, yeah, thanks to the organisers for the invitation. I've not been fortunate enough to dial into the other talks this morning, but I suspect that everyone is equally lamenting not being in Paris during the spring. But anyway, that's where we are. So good that we can still go ahead with the meeting. Just a quick confession before I start. Actually, when I first put this talk together some time ago, I was working for Vertex Pharmaceuticals, and at that point, I would have been able to give some sort of examples to back up some of what I'll talk about, but that's sort of not possible now. But so I'll give a sort of conceptual talk, really, that I hope at least will stimulate some discussion. But the principles that were held dear when I was at Vertex and during my time there are sort of equally held dear by the small company that I now work for, which is Pathios Therapeutics. So just to start off, a well-worn slide, if you'll just indulge me with this one for a second. So this is obviously an analysis of bi-phase development failures for 4 large pharma companies, and you can clearly see that, as we know, that phase 2 failure is at least twice as likely as in any other clinical trial phase. Interestingly, that seems to be true across all therapeutic areas, whether they're main therapeutic areas or more niche areas. But not all therapeutic areas in particular are as bad as others. So autoimmune, for example, is on the slightly better side in terms of likelihood of approval from phase 1 right through to clinical approval, whereas CNS and oncology areas, for example, are often propping up the table. So for CNS, the overall likelihood of approval of a medicine is about 8% from inception of clinical trials to the end of late-stage trials. And if you dig into this further, of course, you know that it's because of lack of efficacy in phase 2. So, some years ago it was mostly PK, but we got pretty good at addressing that with predictive systems and models. But now it's essentially because in efficacy trials, the agents don't work. And there are obviously 2 broad reasons for that. One of them is that targets are either wrong in terms of their choice in the first place, or target hypothesis is wrong, or perhaps the less forgivable reason, which is that the hypothesis was never tested at all due to sort of lack of competent biomarkers, etc. And that obviously focuses the mind on 3 key points, which is picking good targets, developing good competent translational strategies, and designing effective early clinical trials to show that we're engaging the target and the mechanism of interest. And I'll say a little bit about each of these, since it turns out really the requirements for getting each of these right is essentially the same. And that's a principle that I hope to draw out during the talk and say a little bit about. And yeah, I'll also touch upon why certain therapeutic areas perhaps look different to others, and introduce this concept of the target disease trajectory, and talk about why that path from target to disease symptoms is sort of really critical in determining good projects to work on, and how to, how at least I think it's important to prosecute certain types of projects. So just to define these concepts in a little bit more wording. So for a good target, we might think of something where there's a really good link to human biology, where the basic function is well understood, and essentially the link between target and some relevant phenotype is well defined. For translation, we're looking for primary preclinical measures that are quantitative translational readouts, things that we can measure in different contexts in cells and animals, and put good fidelity on those measures. Whereas in early stage clinical trials, we're obviously looking for proximal mechanistic biomarkers, PK/PD biomarkers, essentially, that give us an early indication that we're likely to see efficacy in later trials. And what's really key here, of course, is that actually these things are essentially all of the same thing. And what they're basically referring to is a relationship between target cell and multicellular biology, so the proximal downstream biology that links target perturbations to certain types of outputs. And the key is, of course, the ability to measure these across multiple platforms and contexts, be it in relation to natural variations in biology, so perhaps genetic variations within cells and animal systems, and then ultimately in early clinical trials. But essentially, it's referring to exactly the same thing. And another way you can portray this, of course, is like this. You can think of this as perhaps a dose-response relationship between a target perturbation and its proximal and downstream biology. And so when you apply this to target validation, we're obviously looking for, again, natural variations in target activation. Experiments of nature, usually based on genetics, but not always. We want to look for this in humans, ideally, not in other types of systems, since that's where our drugs are ultimately will be used. In terms of the sort of drug discovery cascade, we're looking to generate agents, obviously, small molecules, large ones, DNA, RNA, where we can manipulate that target effectively and essentially recapitulate that target validation dose response in preclinical system cells and animals, etc. And then in clinical trials, utilize those measures we've established to use competent agents to establish that dose response again and close the loop. But essentially measuring, again, that plot on the left in all of these different contexts. And it's obviously very simple and easy to state, and nothing new here essentially. But I think it is surprising how often perhaps certain types of biology and approaches seduce us away from these sort of basic tenets of what we're trying to do. And just another point to make at this point is that at the moment I'm not really talking about diseases or symptoms, and that's very deliberate. I'm just talking about biology, and I'll come back really later to why that is at the moment. Just talking about sort of systems biology, proximal measures to target perturbations. And so just to give a few classic retrospective examples in some cases that you're probably very familiar with, where this sort of plays out quite nicely. So KCNJ11 is a gene which encodes potassium-sensitive ATP channels, and they're highly expressed in pancreatic beta cells, and therefore, if you get mutations that cause a loss of function in these channels, you can get hyperinsulinism through a sort of overactivation and excitability of beta cells, and then certain types of gain of function mutations that obviously cause the opposite, which is diabetes. The poster child of sort of genetic validation is obviously PCSK9. It's a protease that cleaves the LDL cholesterol receptor. So you get rare gain of function mutations that lead to hypercholesterolemia, and then sort of more common variants, which leads to either, in the main, small reductions or sometimes elevations. And these are linked to cardiovascular events, of course. And then just another example in terms of the JAK-STAT pathway, we know that loss of function mutations lead to immunodeficiency and frequent infections, whereas gain-of-function mutations cause autoimmune disease or are linked to autoimmune disease and increases in cytokine signaling. And of course, as you're probably already guessing, the reason I'm showing these is because they quite nicely predict or correlate very well with drug effects. I don't really need to go through these in lots of detail. Obviously, you can identify openers and blockers of potassium channels in the pancreas, and they have opposite effects on insulin secretion. There are monoclonal antibodies against PCSK9, which can lower LDL levels, and obviously JAK1 inhibitors are starting to show efficacy in autoimmune disease. And so this all looks good, and you can summarize what's attractive about these targets, and you can identify this ability to show that there are very very clear measures that are proximally related to the target and target modulation that are what I'd like to call perhaps meaningful quantitative entities, at the cellular and multicellular level. So for KCNJ11, you can think of insulin levels, PCSK9, obviously cholesterol levels, and perhaps JAK-STAT. You can think of T cell proliferation, cytokine signaling. And what's also really important is that you can measure these equivalently in different settings. So be it in just looking at natural variation in response to different genetic variation, in vitro, in vivo, in preclinical settings, and obviously later in clinical trials. And then you can expand these measures to look at their relationship between perhaps regulatory disease endpoints in sort of subsequent late-stage trials. And at the core of this really is systems biology. So the ability to link target manipulation and variation with proximal systems or cellular level biology phenotypes. And these are obviously hand-picked examples, and we can see why this all sort of looks quite nice. But why are these proximal measures so important, and why is their relationship to disease states so important and so valuable? And a way I like to think about this perhaps is to visualize what I call a target disease trajectory and think about its corresponding complexity. So in terms of target validation, you can think about perhaps there being a target and some perturbation of that target, which is shown on the left. And then there's a trajectory from that target to its influence on disease perhaps in different people. And naturally, as you sort of traverse this trajectory from left to right, there's an obvious increase in complexity and divergence in time and space, which influences, the ultimate endpoint on the far right there, which we can call, for example, human disease, and then maybe other processes and other target variations that start to increase the noise as you go from left to right. So therefore, the highest fidelity measure of target perturbation is clearly as far to the left here as possible, even though the disease state, as we think of it in terms of symptomatology, is more towards the right here. And the obvious problem, of course, is that these measures on the left are much harder to come by and more difficult to measure, potentially more invasive. You can also think of this in terms of just sort of basic translation and apply the same framework. So, for example, you could imagine that you've got a measure that you want to look at in cells, in animals, and then try and measure that similarly in humans in clinical trials. And of course, the measures here on the far left, which are more tightly related to the target biology, are going to look more similar between all those settings than ones on the far right here, where you're having to deal with all sorts of different variables that start to impact that trajectory from left to right, not least of all including the difference between an in vivo and an in vitro context, and perhaps differences between species, i.e. preclinical species, and ultimately doing work in humans. And of course, you can continue to look at this in terms of early clinical trials. So again, these measures here on the far left were much more tightly related to target perturbation and allow or enable you to look more clearly at of whether that target has been engaged and is being manipulated in a mechanistic way that it's meant to be, as opposed to these measures on the right, which, even though they have relevance to the disease state perhaps more closely, are much less closely related to the target biology and offer a much less or lower fidelity measure of target perturbation. And of course, as I mentioned, up to now, I've not really talked about diseases or symptoms. I've just focused on this sort of target biology dose relationship, and that's been very deliberate. Now, of course, at some point, you do need to connect the dots between these target perturbations that are proximal to the target and ultimately disease measures which are relevant for regulatory endpoints and ultimately drug approval. And these dotted lines very simply represent those two scenarios. What's quite clear, though, is if that measure is well understood at the target validation phase, then there's no real reason to need to measure that again until a clinical trial phase. The translational mechanics, or the under the hood mechanics, if you like, of creating a drug which manipulates that target and then works in a human setting, is really served by this sort of machine level understanding of the target, which goes across the green lines there in the middle. The other thing to note, of course, is that these dotted lines here cover a multitude of potential complexities. So in some cases, and I'll show for the examples that we talked about already, this is relatively simple, whereas in other cases it's far from it. And I think there's one message that I'd just like to perhaps portray, is that having a good understanding of what that complexity looks like in time and space, and a realistic appreciation of that, is perhaps the key in terms of perhaps where to put investment in the drug discovery business. So again, in terms of the three targets that I showed you as the nicely handpicked examples, we can look at proximal measures of target perturbation, which we know are very clearly related to those targets in different contexts, be it through genetic variation or in response to drugs. And we also know that they're quite tightly linked then to disease symptoms and things that we would measure from a regulatory point of view. And therefore, that target disease trajectory, at least in these cases, is relatively simple and straightforward. But that's obviously not always the case. And just as an example, therapeutic area that I wanted to touch upon, which really I think highlights this quite nicely, is neuroscience or CNS drug discovery. So an area that I worked in for a long time, lost a lot of sleep over, and spent a fair bit of time thinking about the reality of being able to sort of address certain types of neurological disorders and our readiness and knowledge level, really, where that sits in order to do that. So as one example, you could think about epilepsy, and this is interesting because, of course, there are relatively proximal measures to target biology that we can get our hands on. So if you imagine a variance in an ion channel that causes changes in neuronal excitability, we can quite quickly get to things like invasive and non-invasive EEG that relatively easily index those sorts of changes, at least relatively speaking, in terms of the CNS field. And then obviously seizures are essentially a manifestation of hypersynchronous brain activity, again, which is measured to EEG. So arguably the target disease trajectory there is relatively straightforward, at least in terms of the neuroscience setting. And then it's probably no wonder that unusual for neuroscience animal models, epilepsy models have been shown to have quite good translational value, and it's an area where there are various medications available with clear links, actually, at least in retrospectively, to human genetic variation. You could probably put sleep disorders in a similar bucket. So we know the sorts of circuitry involved in sleep, and that's relatively well described and easy to measure via EEG states. Again, a form of hypersynchronous activity to some extent, and not too dissimilar from epilepsy, a more physiological form of synchronous EEG and electrical activity. And then it's quite straightforward to link those different types of EEG states to, if you like, diseased forms of sleep, such as insomnia or narcolepsy. And so the disease trajectory, the target disease trajectory in this case, is also not particularly complex, relatively speaking. And there are proximal measures you can get your hands on. And then as you start to go into other CNS disorders, this looks a lot less straightforward. So, for example, in psychiatric disorders, it is much more difficult to identify proximal markers and target perturbations, which we can measure well, understand well, and then ultimately link to psychiatric symptoms that are relevant to regulatory approval and in terms of diagnosis and assessment of mental health. And arguably, things get even more tricky when we talk about neurodegeneration. We know that there are targets that lead to disease, and there are well-linked genetics to things like Alzheimer's disease, ALS, and Parkinson's. But understanding what they do in terms of the proximal biology and the downstream biology, and especially how that ultimately links to presentation of disease, is really somewhat lacking. And therefore, the disease trajectory here from target to symptoms, and let's talk about maybe cognitive symptoms as an example, is just really daunting and ill-defined. And of course, one of the challenges here is really just being able to identify those markers and measure them in human biology in an in situ setting. It's just an incredibly challenging thing to do. And so if this is the state of affairs, then it perhaps identifies the target disease trajectory from a translational point of view. You might think, and this is simplistic, that epilepsy, sleep disorders look a little bit like that on the top left, whereas into the neurodegeneration and psychiatric disorders, we are more along the lines of the bottom left. And this equally applies, whether we're talking about validation or then in terms of, looking at the disease trajectory from a translational point of view. That lack of understanding of that target to disease trajectory really just makes everything really difficult in piecing together that thread of measures across these different domains of firstly, human biology in response to natural variation in preclinical settings and ultimately clinical trials, that link between changes in the target activity and some proximal biological measure that we can relate to more disease relevant symptoms. So translation and drug discovery here just becomes incredibly challenging. And you can look at it in terms of other disease areas. So just a few that I've pulled out that we sort of care quite deeply about at Pathios. So in terms of autoimmune diseases, perhaps things look a little bit better. We can link target perturbations very clearly to cell signaling events and cytokine release. And we know quite well how that links to different types of inflammation. In oncology, you can imagine that the link between targeted therapies that go after perhaps certain mutated kinases and cell signaling, cell death, proliferation is reasonably well described in terms of the link to ultimately tumor growth and volume. Not simple, but more simple than, for example, something like neurodegeneration. Whereas in immuno-oncology, that trajectory is a little bit more complex. We're obviously looking at the interaction between targets in the immune system, activation of that immune system or release of the brakes on the immune system perhaps, and ultimately impact on tumors. So that's a little bit more involved and more complex than, for example, certain other types of oncology approaches. But still, there is a lot in there that we do understand, and that's been shown to bear fruit in the clinic in obvious approaches, and maybe somewhere we can expect to see success in the future as well. So yeah, again, just being a little bit flippant in terms of identifying how these look. Perhaps there's a little bit more complexity here in the bottom than the top, but essentially there are things that we can get a handle on here, and it's not completely impossible and as difficult perhaps as some of the examples in neuroscience that I showed you. So just to sum up quickly, obviously in terms of identifying targets, we want to start with human biology, identify targets with that clear dosage effect on human phenotypes in response to natural variation, and then prioritize phenotypes that we want to look at that are quantitative and can be measured at all stages of drug discovery, right through from target validation through preclinical systems and into early trials. And we're looking for a well-defined target disease trajectory, so one where those proximal measures have a nice correlation between perhaps more clinical and regulatory endpoints. And I guess the warning is to beware of complex, ill-defined target disease trajectories unless we're prepared to invest in and identify these sort of quantitative and target proximal human measures along those trajectories. And I think that the challenge there is, the seduction of certain types of biology can be very strong, and the unmet need there can be very strong. So we need to be smart, I guess, in how we think about those situations. And I guess what this focuses the mind on really is there are two types of technologies that are really key to modern drug discovery. One of them is in a situation where we know there's a good target and all of these boxes are ticked. It's really finding the technologies to prosecute those targets and expanding that toolbox, and be it certain forms of advanced small molecule discovery, perhaps structure-based, fragment-based, or then perhaps using larger molecules, be it antibodies, ASOs, gene editing, etc. And then the other set of technologies really are around better ways to measure human biology. And I think the key word there is human. And it's very obvious to see why that's difficult in neuroscience. Those measures largely would need to be invasive given the technologies at the moment. But I think where our ability to crack hard diseases will come from is not by looking in cells, not looking in animals, and not even trying to find better agents to address those situations. It's probably being able to better measure those diseases in a human setting with sort of tightly correlated quantitative measures to the targets of interest. So yeah, that's it. So thanks very much for your attention. Happy to take any questions.